Good afternoon, everyone, and welcome to the Institute of Advanced Studies. I'm Ksenia Chmutina, I'm the director of the IIS, and we're here today to welcome our fellow, Dr. Adam Heathcote. It's a hybrid seminar as always, so we have a lot of people in the room today, which is absolutely fantastic. We also have people online. Our talk will be recorded, but we are not recording questions and answers. The Institute of Advanced Studies is really home to transdisciplinary conversations that we have here at Loughborough University. And it's wonderful to host so many fellows who really cover the most exciting topics that we have to discuss. And so today I think will be a fantastic example of that, and Adam is going to talk to us about lakes, history, and ecosystems. And I've kind of had a sneak preview of the slides, and they're beautiful, so it's going to be a lot of fun. Without any further ado, I'm going to pass over to Professor John Anderson, who will be introducing Adam.
Okay.
Over to you, John.
Right, brief introduction to Adam. So we were discussing this morning how long we've known each other, and Adam recalls the fact that I asked a question at the ASLO Limnology and Oceanography meeting in Nice in 2009. It was a very polite question, of course, and it was about eutrophication. But in fact, I recall I actually moved, my first encounter was when I had a paper from Ecosystems to review, which was eventually published. And that is actually some of the work we've been doing since on carbon bearing. Adam did his PhD with John Downing at Iowa State. And after that, he went to work as a postdoc with Paul Del-Giorgio and Yves Prairie at the French University in Montreal. They're three of the probably most preeminent limnologists, and one leads to the question of why is he working with me and been working with such other people. So after Montreal, he returned to the US, but this time the state north of Iowa, Minnesota, as a postdoc. And now at the site, he was working at the Science Museum, well, went back to the Science Museum. And now he is actually the director of the research station on the St. Croix River, which is part of the Science Museum. And he leads the lead-210 dating facility there, which is, I think, one of the world's great lead-210 facilities. But he started as a diatomist, but he considers himself a limnologist. But his key skill, I think, is his ability to handle big data and statistical skills. Which is really what we're going to be, I think, we're hearing about today. And finally, I would just say he's got a lot of money, research money, NSF and state funding, and we've been together in Greenland. And I think with that, that's enough. I think.
Definitely,
you,
Adam.
Adam.
Thanks. I don't know if I should stand or sit. I'll stand.
Oh, you're able to sit. Okay.
If I have to sit, okay.
Okay.
For the camera. It was not an option. Okay. Where should I look?
Oh, that's fine.
Okay, all right. Well, thank you very much for that introduction, John. So what I'm going to share with you all today is a talk that I haven't given before, so I appreciate your grace. I wanted to put together something new for you all. And so I mixed together a couple of different stories, all loosely tied to this theme of using lakes as sentinels of global change. I like diatoms to DNA because of the alliteration, but I am actually going to talk about both of those things as well as some geochemistry. But before I get started, I wanted to extend a sincere thank you to Loughborough University and the Institute of Advanced Studies for inviting me here, and especially for the staff who have helped with all my accommodations. It's been very lovely so far on this trip. And then also to my good friend and colleague, John, who's been my host, and who you can see here. We're probably discussing the next great paper here on the shore of a lake in Greenland. Or maybe we're just, talking about football scores. I'm not sure. I can't remember. But oh, birds, actually. Yeah, it's probably birds. So since I'm the one keeping you from lunch today, I'm going to give you a overview of the timeline so you can keep track of how long you have left before we can eat. So we'll start with diatoms, a chapter on nonlinear responses to climate change in the Arctic, a lot of which people at Loughborough University have actually been involved with. I will focus mostly on the algal portion, which I helped with. Then we'll move to something that I'm actually working on as part of the fellowship here with John, which is a pan-Arctic synthesis of carbon sequestration in lakes. And then finally, I'm going to end with a new realm to paleolimnology that I've been moving into recently, which is the use of sedimentary ancient DNA to reconstruct organisms that aren't often well preserved in sediment cores. Specifically, I'll talk about cyanobacteria or blue-green algae. But this same technique can be used for a whole host of micro and macroorganisms. So I want to mention that this work in Greenland was a huge interdisciplinary and collaborative project. I was only a small piece of this. This particular part of the study was led by Dr. Jasmine Saros at the University of Maine. So you can see on your right there on the. And so I'm just a little bit of background on why we were working in Greenland in the first place. So as most of you probably know, there has been rapid warming in the Arctic. This is an annual report card published by NOAA, which shows the relative warming each year and keeps a running 30-year average total. From this, we can see that most of the Arctic began warming almost 50 years ago in the 1970s. However, it's now warming about twice as fast as the global average. So if you can't see the text there, the red is the red is the Arctic mean annual temperature anomaly, so the difference from the mean, and the gray line there is the global average. And so this means that, for example, the 8 warmest years on record have occurred since 2016 in the Arctic. And in fact, last year, 2023, was the warmest summer on record, which is that figure on the right there. However, things were a bit different in this area of Southwest Greenland for various reasons. And actually in the mid-20th century, so about the same time the rest of the Arctic began warming, this area of Greenland was actually in a cooling or static phase. So when we ran a breakpoint regression, which looks for nonlinear change of a time series, we see a reduction in the mean temperature after 1960. So again, when the rest of the Arctic is already warming, and then stabilizing and then jumping up to increase nearly 3 degrees after 1990. So this is very different than what we see in the rest of the Arctic. And Greenland isn't the only place that was like this, but it's one that we've worked in. And so the opportunity here is this allows us to compare models, paleo records, as well as monitoring data, because there's quite a bit of on-the-ground monitoring that happened prior to warming in this case, whereas the rest of the Arctic, that may not be the case. And so it's a broad opportunity to look for nonlinear environmental and ecological responses to warming and how we might predict those happen in the future in other parts of the Arctic. So to orientate you a little bit into what Southwest Greenland looks like, it's mostly dwarf shrub tundra, which is what you can see us walking through there. So that's predominantly Bechulinana or dwarf birch. You can see this is in the fall. It's just starting to change colors. And then it transitions to steppe as you get closer to the ice sheet, which is in the foreground there. In the background, you can see that's the Russell Glacier coming out to the outwash. So this land is dominated by glacial processes, both the ice sheet itself, as well as the Sander or the major outwash, which flows directly through, kind of bisecting the area we work in. You might think we're just stopping here to take a break, but notice that John has his map out, so this indicates that we're actually lost. So the area we're studying on here, this is just a map. You can see Kangerlussuaq. This is where the airport is. It's a very small town, but it happens to be where the American Air Force Base was during the Cold War, and it's now a Danish military base. And so it's the only place where you can land a 747 on this part of Greenland. So it's a convenient place to work. And as I mentioned before, this was a broadly interdisciplinary study by a lot of people. You can see we looked at everything from the phenology of the plants to the outwash of the glacier, the discharge, as well as the movement of dust. But I'm purely going to focus on this one little piece here, which are how the lakes themselves and the algae within them responded over this time period of great change in temperature. So the data that I'm going to be presented is fairly routine for most paleolimnologists. So we have basic ice out data. So this was monitored from the largest lakes of this region in Southwest Greenland starting in the 1970s. So this is the time when ice basically breaks up on the lake each year. And then we also looked at diatoms, which are this microscopic algae, which has a siliceous glass wall, which is perfect for paleolimnology because it's well preserved. And also they're intricately designed, which means that we can identify them to species. And they have various tolerance ranges for different geochemical measures of interest. So they're a perfect kind of indicator species. So we counted these diatoms to species in various sediment cores. As John mentioned, I run a radiometric laboratory, and so we also dated these sediment cores using lead-210, which is a naturally occurring radioisotope, which allows for about 2 to 10 year dating resolution over the last 200 years. So it's perfect for this period of the Anthropocene. We kind of summarized the diatom species data using kind of standard ordination techniques. Here I'll be showing principal components analysis, which is just a decomposition of highly dimensional data into a single dimension so that you can see broad changes in many different species at once. And then, one of the unique opportunities we had here is that because the various researchers who were on this project, John included, had been working in this area for multiple decades, we actually had monitoring data as well as algal extant data that was collected both before and after this warming event so that we could compare those species that were there. So more than just paleolimnology, actually comparing monitoring. So the first time record here is that ice out record. And so you can see on the y-axis, the vertical axis is the ice out day of year. So the warmer it would be, you would expect ice out to be earlier in this region. And we see almost a week earlier. And I'll mention that that breakpoint is, if you can't see it, is right about 1995. So it lines up almost perfectly with that jump up in temperature. And that led to a nearly 1 week earlier ice out time, which may not seem like a lot of time, but remember, ice-free days are very short in the Arctic. So this is a significant amount of time that these lakes would be exposed to the atmosphere. And responding to that, we looked at the various species of diatoms. So this is just the first axis of that principal components analysis for 3 different cores. And what we see is that major turnover starts right at that breakpoint and for at least 2 of the 3 cores and continues afterwards. So this is basically the changeover in different species, often called beta diversity in the ecological literature. And so to kind of zoom in on what that meant for those species, or what is the actual ecology of those diatoms that are changing, we looked at this before and after these surface sediment samples. So this isn't looking at cores, this is actually looking at what was living in these lakes before and after from collections. And we also have monitoring data from the lakes themselves. And so we saw, this is just a pre versus post comparison. So this is a comparison of samples collected in the early 90s to samples collected in 2013 on the same set of lakes. And we see that temperatures increased, water temperatures, the dissolved organic carbon, so that's the staining of the lakes, decreased, which also resulted in water clarity itself increasing and a reduction in nutrients in some of these systems. And so how this was reflected in the diatom community, the first thing we noticed was a decrease in the planktonic to benthic diatoms. So these are diatoms which are free floating. Those are the planktonic diatoms in relation to diatoms which are either living in the mud itself or on plants. And so what we see is these planktonic diatoms decrease. And one of the causes for that is the dropout of this specific cyclotoid species. So this is a centric, a round diatom called Discostella stolidura. And this is a known indicator of warming because it resides on what's called the thermocline. So that's the transition layer in lakes between less dense water and deeper water, which is colder and more dense. And so they actually live in this area called the deep chlorophyll maximum. That's what DCM stands for. And as ice out becomes earlier and earlier, that thermocline becomes deeper and deeper, and they are no longer able to photosynthesize at that area. And so they often will start to disappear. In contrasting to what we're seeing in those planktonic diatoms, we're seeing this extensive increase in the benthic diatoms. So these are the diatoms living on the sediments, or the algae themselves living on the sediments. You can see lots of examples here. These are actually some of the cyanobacteria nostoc, which probably has millions of diatoms living in and among it here. This is often called benthification. So this is a change from a plankton dominated system to a benthic dominated system in terms of primary production. And what we see in the diatom records is an increase in all of these high profile algae. So panel D there, if you can't read, are just things like colonial algae. They're high profile algae, so algae that are growing on things like mucus stalks, things like Gomphonema, if you're familiar with any of the genera of diatoms. So big things that are growing on plants and growing out from the mud itself. And the really little tiny things, the things that might live within sand grains, are disappearing. And we're also seeing a diversification in terms of the size class of these organisms. So the middle panel there shows in white what the size distribution of diatoms were in the 90s versus today. And so we see a decrease in the number of these really small ones and a broadening, a diversification in the size distribution. And this trend is most distinct in the deepest lakes. So if a lake was already very shallow, it already probably had benthic production dominating the entire lake area. However, if it was deep, it has more area of which you could have expansion of this benthic area. And so all these changes to primary production could obviously alter carbon dynamics because the algae are the primary CO2 fixer in aquatic ecosystems. And so this could lead to questions of how is this affecting the geochemistry? And so that brings us to the second section here, which is carbon sequestration in lakes. And again, this is a broad collaboration. As I mentioned, this is a pan-Arctic synthesis. And so you cannot do this type of work without being collegial and reaching out to colleagues to get them to share their data and share their ideas. So this represents a whole group of people here, including John. And some of these figures, I'll have to say, I developed on John's patio yesterday. So that's how new some of these are.
Was that before the gin and tonic?
No comment? Well, you be the judge. So why do we care about soil carbon or carbon in the Arctic? Well, there's a huge store of it, about 1,700 petagrams, and a petagram is 1 billion metric tons. And it's also within this very dynamic area where you have deepening of the active layer as permafrost thaws. And so there's a high potential for nutrient transfer for nutrients that have been locked up for almost the entire of the Holocene. This is just the same map, but just showing soil organic carbon content in terms of density in kilograms per meter square. And so the question then is, what is the future fate of this carbon, as well as carbon that could be fixed directly from the atmosphere as CO2? So why do we care so much about Arctic lakes? I care about lakes in general, but I'm going to convince you they're particularly important in the Arctic, and that's because they're very, very densely distributed. So most of the Arctic is relatively rich in glacial lakes. So these are glacially scoured lakes. Between 6 and 15% of land area, and for context, the global average, that's about 2 to 5 times the global average. So much more lake dense than the rest of the world. This is just a map of, again, the Kangaroo Loose Lock area showing the density of lakes there. However, there are areas within the Arctic which are even more dense, and these are active thermokarst areas. So these are lakes that are actually being created by the thawing of ice wedges within the permafrost. And some of these areas can have lake densities greater than 20%, which could be something like 10 times the global average. And also in these areas, it's important to point out that that lake area is highly dynamic. So with permafrost thaw, you get expansion of lakes as smaller lakes merge into larger ones. And then you also get catastrophic drainage of lakes at some point where they will drain out into the ocean all at once in major events. And so the actual transition between those two is actually an area of active research, whether we're gaining lakes or losing lakes on average in the Arctic because of this process. So again, I'll espouse the power of mud. Lake sediments are a major organic carbon store in general. In particular, we're interested in the Arctic. And these sediments provide a record of change. And importantly, we can actually date the timing of these changes using either radiochronology, lead-210. And here's an example where you can actually see that chronology. This is a freeze core of varved sediments. And so those are actually annual laminations within the sediment core. So the goal of this is let's measure the rates of past carbon production in Arctic lakes, as well as the transfer from soils and from the atmosphere. We're going to try and answer 3 questions. How does carbon burial change in a warming Arctic? What is the fate of terrestrial carbon from thawing permafrost? And what can we predict for the future? So as I mentioned, we assembled this synthesis. This included cores from Greenland, from the Ural, Siberian, Kamchatka, from Arctic Manitoba, the Yukon and Alaska in North America, as well as Fennoscandia and Svalbard. All lakes are within this permafrost zone, either the continuous or the discontinuous permafrost zone. So we're defining Arctic loosely here. So we're not just saying north of the Arctic Circle, but we're saying lakes that are within this permafrost zone. And of course, the Arctic is not homogeneous. There's many different vegetation types across the Arctic. I introduced you to the dwarf shrub tundra in Greenland. There's also tussock tundra. This is an example from Alaska, as well as transition. So you can actually have some trees in parts of the Arctic where you're transitioning from taiga to tundra. And all of these different vegetation types are represented in this data set. There's a wide range in temperatures. Of course, they're all cold, but their annual temperature ranges from -13 Celsius to right around 0. And precipitation is even more variable. So you go from nearly desert-like, less than 200 millimeters rain per year, to over a meter in some of the maritime Arctic habitats. And then another measure that will become important that I'll introduce you to is the measure of greenness. And so this here is summarized from satellite data, and it's a measure that's called, I'll call NDVI, but it means the normalized difference vegetation index. And it's basically looking at the ratio of near infrared spectra to red spectra to be able to look at how green things are. And this is all from remote sensing satellite data. And then finally, I'm going to separate those thermokarst lakes. So that was that highly dense lake landscape, because we already know that those are acting very differently, unlike the rest of the glacial lakes, which were formed from the scouring of the ice sheet or the leaving behind of ice blocks. These are actually being formed by the thawing in place of permafrost through the process you can see at the top there. And we know that these have extremely high carbon fluxes, and we hypothesize this might be due to the fact that they're very active, and there's lots of lateral transfer of soil organic carbon into these systems as the permafrost thaws. So right off, here's all the data. You don't need to know anything else. So we see significant increases of carbon burial. So you don't worry too much about this. It's just all to show that we have lots of data points and that many of them are going up. We see a wide range though. So from 3 to 10 grams of carbon per meter square per year. So almost a threefold change across the data set. And a variability across regions. So here they're just colored loosely by vegetation types. And then we've also separated out those thermokarst lakes, which you can see sitting on the top in blue, much higher than the rest of the systems. And also we can see that this increase predated at least what we think of as warming in the Arctic. So we see actual increases when we compared pre-1900 carbon burial rates to those from 1900 to 1970, which is probably a bit of a response from this part of the world coming out of the Little Ice Age. And so what's driving this change? Well, here I've included the Arctic lakes in blue. I've separated out the thermokarst lakes in red because they're acting a little bit differently. And then I've also lopped on lakes from the boreal taiga because that's kind of the natural extension of this data set. So this is work that John and I did previously, so that you can kind of see the transition here. And so what we see is there is a significant relationship to latitude. Warmer latitudes are fixing more carbon than colder latitudes. However, this drops out when we actually look at temperature. And a lot of this is to do with these thermokarst lakes acting as outliers, burying much more carbon than we would expect given the temperature. However, if we change this around and look at the vegetation productivity or the greenness of the environment surrounding these lakes, we see the strongest correlation. So this is either to do with an indirect relationship, so fertilization, which is being reflected by the greenness of the watershed, but is in lake itself, or that carbon that's being fixed by the watershed being transported into. We can't say which of those two it is, but it's by far the strongest significant relationship to explaining this change in carbon burial. And then if we look at sources, so here we're looking at the ratio of carbon to nitrogen, which kind of tell you how terrestrial versus aquatic a sample is. So if it has a really high carbon to nitrogen on the right of that plot there, that's probably mostly terrestrial because nitrogen is preferentially used by microbial activity, and vice versa. And then we're also looking at the delta 13C. So this is a stable isotope, which as we become more enriched, so we get less negative delta 13C, we hypothesize these are either becoming more allochthonous, soil-like, or they are starting to see the influence of aquatic macroalgae and macrophytes, which have similar signatures to this. So to sum up here, we can see that increasing terrestrial production and latitude correlated with organic carbon burial. The highest rates of burial are in thermokarst lakes, with some rates similar to hypereutrophic lakes. So here I've summarized all of the data. In green are the non-thermokarst lakes, in blue are the thermokarst lakes, and then I put 3 horizontal lines. The first one being the boreal lake average, the second one being the thermokarst lake average, and the top one there being hypereutrophic lakes. So these are like highly impacted urban or agricultural systems. This is how they're functioning. And you can see some of the thermokarst lakes are already approaching those levels. And if we sum up all this information, so we take all these cores and we integrate all that carbon burial and we multiply it by the total Arctic lake area, we can kind of see how much is being buried over time. And I've binned it here into decades. And so we can see that from 1900 to when our data set stops, about 2000, that the difference between the top of that first bar and the top of the last bar before the red dashed vertical line there is about 9 teragrams per year difference. So we're fixing now about 9 teragrams or 9 million metric tons more carbon in just Arctic lakes than we were 100 years ago. And we predict, using the extension of this general additive model here that we've ran, that could go up by another 10 teragrams per year by the year 2100. And this is highly dependent on this concept I talked about in terms of whether thermal karst lakes increase or decrease in total area. And that's what that error bar represents. That's plus or minus 30% lake area for thermal karst lakes alone. And so as I mentioned, that's an open question. We don't know the answer to that. The take-home message here is that since 1900, these lakes have buried about 1.2 petagrams of carbon total, which is about 54 total petagrams stored in Arctic lakes just in the last 100 years, which is about 50% of the previous estimate of the entire stock of carbon in all lakes in the world. So finally, we're going to go to sediment DNA as promised. And this was again, a collaborative project by myself and collaborators from both the University of Ottawa in Ontario, Canada, as well as the University of Regina in Saskatchewan, Canada. And so we'll switch gears here a little bit. We just spent a while talking about the Arctic, and now we're going to talk about more temperate systems. And we're going to be talking about the issue of eutrophication. So this is a natural process which occurs where lakes become more productive over time, over many thousands of years in natural systems, but one that we have rapidly accelerated as humans to what we now call cultural eutrophication. So this is accelerated productivity being driven by the increase in export of nutrients, either from agriculture or urban environments. And so paleolimnological evidence has shown that this has largely increased across Europe and North America. This was a study by one of the collaborators on this study, Zofia Taranu, who's now at Environment and Climate Change Canada, showing data from lowland Europe as well as lowland North America and alpine systems. And what we're looking at here are pigment concentrations. So these are fossil pigments which are unique to this group of algae, cyanobacteria, and showing how they increase over time. So this is another type of proxy we can use. You know, when we look at diatoms, we look at their fossil glass cell walls themselves. Here we're looking at special accessory pigments that are produced only by this type of algae. And so there are other ways we can reconstruct cyanobacteria. They do produce resting cells called aconites that are stored in sediments, and so you can count these. However, there are only certain genera produce them. So two examples that do are Anabaena and Aphanizomenon, but it leaves out large groups of these cyanobacteria if you only count these aconites. And you can also measure the toxins they produce. So one of the reasons we're interested in cyanobacteria is that they produce potent liver and neurotoxins, which are harmful for both humans and wildlife. And so we're interested in tracking this through time. So when we talk about the concept of harmful algal blooms, the reason they're harmful is because of this production of toxins. So the use of DNA offers a complementary approach to these previous ones I mentioned. It doesn't replace them. It's just an additional one that can be used. And how this works, there's a couple of different ways we can do this, but what we're doing is we're actually extracting DNA from different sections of a sediment core. And we're measuring it in a couple of different ways or quantifying it. So one way is by looking at specific primers. So these could be primers that are specific to a taxonomic level. So it could be a primer that is only present in cyanobacteria. It could be a primer that's only present in the genus Microcystis. It could be a primer that is related to a gene that regulates toxin production. So depending on what your question is, you can adjust the primer you use if there is a unique genetic or a nucleic acid sequence that corresponds to this. And so this is called, when you do this quantitatively, this is called quantitative PCR, or ddPCR is a more precise method that's being increasingly used, which is called droplet digital PCR, which basically takes all the pieces of DNA, puts it into tiny oil droplets, and then measures the amount of it in each sample. And this is just an example from a study which used this. This was kind of a proof of concept. So they did this in the experimental lakes area in Ontario, Canada. So this is where they experimentally eutrophied a lake back in the 70s. And they went back, took a core, measured the DNA there to see if it faithfully tracked that symbol of known eutrophication. So you can see on the left is a reference lake within the experimental lakes area, and on the right are lakes where they did the nutrients addition. And there are various primers there related to different types of cyanobacteria. So that's the first thing you can do. The second thing you can do is what's called high throughput sequencing. And so this is actually getting down to what are the organisms that are there. And so this can be done in a bunch of different ways. What I'm going to be presenting is often called shotgun or amplicon sequencing. And so this is kind of looking at short reads of DNA, which are unique to different organisms, which are all stored in the library somewhere. So you can compare your nucleic acid sequences to a library to figure out actually what species or even what strain in some cases of cyanobacteria is present through time. This is an example from 10 European lakes, para-alpine lakes, where you can see you don't need to worry about any of these names. All you need to know is these are all different strains of cyanobacteria. And starting on the bottom are sections taken from the early 1900s going into the 2010s. And so it's basically a heat map of where these different strains start to occur and where they're most dominant. And so we've actually combined these two approaches for this study. So we have both the quantitative side using the ddPCR, so looking at specific genes and genera. And then we've also used this high throughput sequencing to see what all is there. And by doing that, we can see not only how things have changed, not only how the community has changed, but look at actually quantitatively how much there are of different strains. And so to do this, we took a set of lakes from Minnesota. So this is where I'm from. This is the US state of Minnesota. To the north is Canada. To the east is Lake Superior, the largest lake in the world by area. And it's also a kind of unique study area because it has many different types of biomes within the state itself. So in the south are grasslands, tall grass prairie. In the center is hardwood deciduous forest, and in the north is boreal forest, as well as large extensive peatlands. This data set represents diverse types of lakes. So the maximum depth ranges from extremely shallow 2-meter deep lakes to relatively deep 30-meter deep lakes, and trophic state as well. So going from nutrient poor, very clear oligotrophic lakes to hyper-eutrophic, highly impacted nutrient-rich lakes. And land use change runs the gamut as well. So we have agricultural intensification in the prairies, we have urbanization in the metro areas, and then we have relatively undisturbed areas in the north, which is actually one of the most pristine boreal forests in the United States, called the Boundary Waters Canoe Area. It's an extensive canoe wilderness where there's no development. And so, as I mentioned, we did the quantitative PCR as well as the high throughput sequencing, and we threw on pigments as well as kind of an independent measure to see if we were on the right track with these. So I won't get too much into the weeds, into all these details. I'm happy to answer questions that people ask. But in general, we looked at 4 different primers. One was called GLNA, which is glutamine synthetase, which is a bacterial housekeeping gene. It's conserved across all bacteria, so it's a good kind of baseline control for all microorganisms in terms of the DNA. The second one was a primer which is unique to cyanobacteria, so it's only present in cyanobacteria. The third is a primer which is unique to Microcystis. We're particularly interested in this genus because it is problematic. It responds to anthropogenic eutrophication and also produces this toxin, which is produced by this gene, which is the last primer we use. It's called MCYE, which is a microcystin toxin producing subunit. So this species of, or this genus of cyanobacteria does not always produce toxins. It can only produce toxins if it has this gene. And because these are bacteria, they can actually swap genes. So some populations are toxic and others are not. So we're interested both in the increase in the prevalence of cyanobacteria broadly, but also if they are becoming more toxic with time. So these are the results from all 9 lakes in which we did the quantitative PCR on. And what you see here is relatively stable background rates of DNA. So again, on the horizontal axis is just time, going from 1850 to 2000. And on the vertical axis is the actual gene copy numbers of each of those different primers I just mentioned. And so we see relatively stable background rates and then increasing post-1950, the area that we often call the Great Acceleration. So this was kind of the post-World War II boom in North America, as well as other parts of the country. And we also see that most of these increases, so we correlated this bacterial housekeeping gene, so all microbial activity, to just the cyanobacteria primer, and we see that they're highly correlated. So this increase in microbial activity is primarily being driven by increases in cyanobacteria. And those cyanobacteria are primarily this genus Microcystis, and toxicity is also increasing over time, or potential toxicity. So as we get more Microcystis, we get more of this toxin-producing gene, MCYE. So not only are we getting more cyanobacteria, we're getting more potentially toxic cyanobacteria. This is just an example of what this high throughput sequencing looks like. So these are just the 3 lakes in which we did this. And what we're seeing here are the different types, or the different taxonomic levels over time. So again, time on the horizontal axis, and then we're going basically up in taxonomy as we go down. So at the top panel there, we're looking at just rough orders. And so we can see increase in 2 important orders in terms of toxin-producing cyanobacteria, the Nosticales, which include Delicospermum, Anabena, Aphanizomenon, as well as Crococales, which includes all the small little ball-like cyanobacteria, which are really pain to count, but it also includes, importantly, Microcystis. If we break open those 2 orders, which is the 2 bottom panels, so the first one being Nosticales, we see that's being primarily driven by these 2 toxin-producing species, Delicospermum/Anabena. The name has gone back and forth. And the same for Crococales, although we see lots of unknowns. And so unknowns doesn't mean like we can't ever find out what it is. It means that good sequences are not available in taxon libraries. So the really powerful thing about this type of data is that these are all uploaded to a gene library, and someone could come back and pull these sequences in 10 years, recompare it to these improved gene libraries and get better taxonomic resolution as more stuff is added. So the more metagenomes that are added to these public libraries, the better these data will become in the future. And then the other kind of interesting thing that happened here is you can just barely see it, right, this in yellow here. There was a known invasion of a subtropical cyanobacterius, Cylindrospermopsis, in this lake, but we thought it had occurred maybe since the late 2000s. However, from these sequencing data, we can actually see that it was definitely there by the 60s, probably even earlier. And this is interesting because we actually found an amateur phycologist paper that was published in like the Proceedings of the Minnesota Academy of Sciences, where they had microscope images, which they said were this species, but nobody believed them at the time because it only occurred at the time in Florida in the United States. So this basically corroborates that person was likely correct and that it's been here for a while. So, and then we compared this all to a more kind of traditional paleolimnological proxy, sediment pigments. So these are accessory pigments that are unique to different types of algae. That's why algae are called by colors. That's why you have blue-green algae, you have gold-brown algae, you have red algae. It's all to do with these accessory pigments, which is how they divide up the light resource. And so here I'm only showing those pigments which are unique to the cyanobacteria. So if you can't read it here, we have aphanizonophal, which is in nitrogen-fixing cyanobacteria, mixoxanthophal, which is in colonial cyanobacteria, lutein zeaxanthin, which is actually a mixture of blue-green and green algae. You cannot separate these 2 pigments, as well as canthaxanthin, which is in Nosticales, and echinonone, which is in all cyanobacteria. And so we see broad agreement between these pigment data and what we saw across the sediment DNA, which is good because, well, the pigment data were already telling us cyanobacteria were increasing. So this confirms that, but now we know which groups were increasing specifically, and even which strains within those different genera were increasing. The other kind of nice thing that pigments give us is they give us some measure of degradation. So whenever you're using paleo proxies, you always have to separate first order degradation of whatever you're looking at versus actual change. So is it actual increases or is it just natural decomposition of whatever you're looking at? And so we can use this ratio of chlorophyll A, which is a highly labile pigment. So it's quickly taken up by microbes, to its decay product, which is phaeophytin. And so this ratio tells us where degradations might be an issue. And so we can see that, for example, like here, this spike tells us that probably these top 2 centimeters, we don't want to look too much into, or we want to be cautious in terms of interpreting because there's still lots of degradation going on there, whereas it's stabilizing below that. And so this is, you can squint and look at this. I won't go too much into detail here, but this is just to say, this will be familiar analysis for diatomists, but you can use some of the same techniques like constrained clustering to look at how different stratigraphic zones delineate based on the organisms that are there. So you can take this highly dimensional, in this case, 1,200 different amplicon sequence variants were here, and look at what that actually means in terms of community change over time. Similar to what you would do with diatoms. And so when we look at this, we see increases in toxin-producing cyanos in all 3 lakes, I just colored them by either order or genus. So purple are undefined Crococales, blue is Microcystis, orange is Nosticales. So that's again those two toxin-producing genera. Brown is Cylindrospermopsis, and Limnolyngbia is in cyan, which is this benthic algae, which we only see in that very shallow lake in this set. So the broad changes we see are a switch to we see increases in cyanobacteria, but then a switch to diazotrophic cyanobacteria in some of these systems. Diazotrophic cyanobacteria are cyanobacteria which can fix nitrogen from the atmosphere. And so this tells us something about what nutrient limitation and stoichiometry may be in some of these lakes. And the other thing we did is use this new, at least to paleolimnology technique called fuzzy clustering. So this is a technique which has been used a lot in the social sciences, as well as some of the animal behavior sciences. It's called, and I apologize if I mispronounce this, but I believe it's latent Dirichlet allocation, or I'll just say LDA from now on, which takes high dimensional data and puts it into clusters. And how it's different from the one I showed before is that these are fuzzy clusters. So you can actually have the same species in different clusters. And it's instead looking at the relative abundance of those species. So in the simplest example, cluster 1 could have species A at 90% dominance, species B at 10% dominance, and cluster 2 could have the inverse, species B at 90% dominance, species A at 10%. And so what we see is when we reduce this data and we allow this fuzzy clustering, we actually see strong changes around 1950 in all three cases. The time scale is different in these, but I'm talking about this bar here. So this is looking to detect a significant change, a significant breakpoint in the clusters. And this is coincident with the period of intensive agriculture in this area, which led to much of the nutrient pollution and cultural eutrophication. So to sum that up, sediment DNA allows for the reconstruction of cyanobacteria. For 100 plus years at least. There's actually have been several studies which have gone back millennia. Again, this is a very new technique, so the constraints on it are still relatively unknown. And as I mentioned, it gets better with time. Sediment DNA via gene quantification can show us that there's an increase in both abundance and toxin production in cyanobacterial blooms since the mid-20th century. And it also well complements kind of traditional paleolimnological methods such as fossil pigments. So kind of your take-home messages. I know we had three kind of diverse chapters there. The first chapter we talked about rapid warming in Southwest Greenland and how it led to nonlinear changes in primary producer communities, which could be one of the reasons we're seeing large changes in carbon burial in Arctic lakes. And Arctic lakes are globally important in terms of the amount of carbon that they're fixing. And then also I left with this kind of introduction to this technique, sedimentary DNA, which I think is a new and very promising technique in paleolimnology. So with that, I'll say thank you, and I'm happy to take any questions there might be from the audience.